today is protein synthesis translation. Translation goes to 14.4. So, in our journey of protein synthesis, kind of consistent from the central dogma of biology, we start out with DNA, and to decode our genetic instructions, we first go through a process of transcription, which we talked about last time, where we exchange or transcribe genetic information stored in DNA to mRNA. Well, today we're going to talk about that second part of the journey from DNA to protein. And it is creating actual proteins or polypeptides from our mRNA transcript. And this part is called translation. So in a broad strokes overview, this part is what we're going to look at today, from mRNA to protein, or from mRNA to polypeptide. Right? And the translation part happens in the cytosol at the ribosome. So, our transcription happens in the nucleus, and translation happens in the cytosol, in the cytoplasm at the ribosomes. So, translation from mRNA to protein. The goal of translation is to turn the mRNA transcript created by transcription into a polypeptide. So we're actually taking the genetic instructions that were transcribed into mRNA in transcription and turning them into a polypeptide, into a protein. It happens in the cytoplasm, and it involves several different important molecules. One is mRNA, which is familiar from transcription. That's what we made. That's what we turned genetic information into. It's going to require ribosomes, or the organelles that are responsible for actually making the protein. It's going to require TRNA, which is going to be a code reader or decoder, and an enzyme called AMINOACYL TRNA synthetase, which we'll talk a little bit about later on. But the key thing, how does just a bunch of ATCs, U's, and G's in mRNA get translated or turned into a specific sequence of amino acids in a polypeptide? Well, it's sort of like a code, just like the English language has 26 letters, and putting them in different orders spells out different words. An mRNA molecule has a code based on every 3 nucleotides, and a group of 3 nucleotides is called a codon. So here is a codon. On this picture, each different group of 3 colored differently is a different codon. And each codon specifies a certain amino acid that will be incorporated in a polypeptide during translation. So, the triplet code, these codons, their identity determines the amino acids that are getting added to our growing polypeptide chain in translation. So the mRNA codons and the codon wheel are really important. So the main question is, how is genetic information stored in mRNA just by having a sequence of different nucleotides? And how is that sequence actually differentiated into different amino acids? Well, it's these mRNA nucleotides that make up a code. The basic unit of this code is called a codon, and consists of 3 mRNA nucleotides. So, for example, on this strand of mRNA, each group of 3 successive mRNA nucleotides is a codon. Alright? And they code for, or determines, or tells the ribosome that a specific amino acid should be incorporated in the polypeptide. Right? And that's our tRNA molecule that reads this code and adds the correct amino acid that corresponds with this codon to the polypeptide chain. This code is extremely ancient. This code is maybe the oldest example of a code on Earth. It's always the same for all cells in almost all life forms. So because it's so universally used, scientists a long time ago have cracked this code and have made a codon chart like this, or a wheel, that actually gives us the insight into read mRNA. So what does this codon, which amino acid is this code for? Which amino acid this code for? We use this codon chart. So it can show us the different codons possible in mRNA, and then in parentheses it gives us the 3-letter abbreviation of what amino acid is matched up to that code. So if that this codon, these 3 nucleotide sequences is seen in mRNA, it is going to link to this amino acid, and that amino acid is going to be incorporated into the amino acid chain. So for example, in this mRNA, we got ACU, right? We got ACU, which is threonine. That means it's going to be first. So that's going to be our first amino acid. Then next is GGC, which is glycine. So that will be our second amino acid in our polypeptide chain. And our polypeptide chain is just going to be all these amino acids hooked together. And this triplet code, this codon, tells us which amino acid is next in our polypeptide chain. Another important actor is tRNA. So tRNA are the code readers, or the molecules that match amino acids to the codons of mRNA and create the polypeptide. So they bring the amino acids with them, and they match up to the mRNA or assemble in the order of which the amino acid should be in the polypeptide chain. So this is the shape of a tRNA molecule. You can see it's an RNA that is kind of a cloverleaf shape, and with some hydrogen bonds stabilizing it. And it has 2 important parts. At the bottom it has something that's called an anticodon. That anticodon is going to match up, A with U, U with A, C with G, G with C, just like normal base pairing, to the codons on mRNA. And it's going to bring attached up here on the 5 end, right here, has a given amino acid attached to it. Each different tRNA has a different anticodon, and it has a different amino acid. So anytime there's an AAG anticodon, that tRNA will have a certain and always the same amino acid. So we can use them as code readers to put amino acids in the correct sequence. The enzyme aminoacyl tRNA synthetase, it catalyzes joining the correct amino acid with the correct tRNA, so that this code is preserved, that only tRNAs with a given anticodon have a certain amino acid attached to them. So first in translation, we're going to initiate translation. So we're going to start translation with this small subunit of the ribosome. So ribosomes have a large and a small subunit. And it's going to bind to our mRNA strand right here. And then our first tRNA is going to come. And it's going to start at the 5' cap, because we're always going to work 5 to 3, 5 to 3. And it's looking for the AUG codon. So an AUG region on the mRNA, our first tRNA has AUC on it. I was looking for that AUG codon because that is the universal start signal to say start translating here. When it finds that AUG codon, the large ribosomal subunit will come and attach. So kind of sandwich the mRNA in between the large and the small, and it'll take a little bit of GTP to kickstart this process, and we're ready to start translating. Alright? Next comes elongation. Elongation adds amino acids one by one to the polypeptide chain, and it requires energy, as we see in 2 different spots, from GTP. So, as mRNA moves from 5 to 3, so we're going this way, this is moving down, we're going this way, tRNAs match up to the open codon in the A slot, so in the A site. So ribosomes have 3 sites, an A, a P, and an E. So first, a given tRNA matches up with the codon that's in the A site. It matches up, brings with it its amino acid. Second, a peptide bond is formed between the long polypeptide chain that is sitting on the tRNA in the P site and the new amino acid, the next one in the sequence, on the tRNA in the A site. After that happens, the RNA moves down, the tRNA in the A spot goes to the P site. It's called the P site because it's holding the long polypeptide chain. So there is now another empty A site for a new mRNA to move into to uncode or decode the next codon, and the old tRNA that used to have the polypeptide gets kicked out, gets put in the E or exit site, and gets kicked out of the ribosome. It exits. It's kind of a slow moving from out here in the cytosol to A site to P site to E site to back out. Just kind of a linear motion. And then when that's all done, it's time to stop. So elongation continues until a stop codon is reached. So there's specific codons in mRNA that tell the ribosome or the tRNAs to stop making the polypeptide. It's long enough. So some of them are AUG, UAA, or UGA. They don't code for proteins, but for a release factor, a protein release factor. What that release factor does is it adds water instead of a peptide bond to an amino acid. It adds water to the end of this, and it hydrolyzes it, and it cuts it off from the amino or the tRNA, and it floats off free, and the ribosome disassembles, and we're ready to start again. So that's the ending of translation. Last, a little bit of post-translational processing occurs just like transcriptional processing. So many proteins need to be processed before they're active and ready to do their jobs. But firstly, the R groups, like we talked in biochemistry, the R group interactions, they begin to fold up the polypeptide into secondary and tertiary structures as soon as it's synthesized. As soon as an amino acid is synthesized, its R group starts interacting with the other ones, and we get a shape. The shape change. In addition, various enzymes also can modify R groups. They can attach carbohydrates and lipids to them, or they can also cleave a long polypeptide chain into smaller pieces. In addition, some polypeptides that are destined for export through the ER have a signal peptide attached to them. So maybe the first part of the new polypeptide sequence that's being translated is a signal that says that this needs to go into the ER to be transported. And what happens because of that is an SRP, or a signal recognition particle, attaches to that peptide signal and forms a bridge between it and a receptor in the membrane of the ER. And what that does is allows for this amino acid chain, once it's being synthesized, to go right into the inside lumen of the ER for export. And lastly, one mRNA may be translated many times via numerous ribosomes working at the same time. So for example, we might have one strand of mRNA, but several ribosomes attached to it, utilizing tRNAs to translate the whole thing numerous times at the same time. And a mRNA strand with many ribosomes attached to it is called a polyribosome, just like this. Alright, that should be it for today. Let me know if you have any questions.